I think if you zoom out, right, the way that I think about this is if you take a look at increasing levels of abstraction, starting from physics. We've, as a society, figured out how to do all kinds of crazy physics things. Like we can launch rockets and land them very precisely on distant planetary objects. If you take a look at chemistry, we figured out how to make all kinds of synthetic polymers that are never existing, or to the best of our knowledge, never existed probably previously in the universe. And then you take a look at biology, and we're just barely starting to scratch the surface of being able to design and engineer biology. I think that is the next level. It's a very scary thing to be working on, to be fair, because we are biological beings, and if we do something kind of inappropriate, then that would have ramifications for us all. And we don't have right now a good way to kind of control for that. But I will say that it is a really exciting technological frontier to be working on, is this question of like, how do we actually understand life in various ways sufficient to be able to then kind of advance and maybe even one day create life.
Welcome to Galaxy Balance, a podcast at the intersection of synthetic biology, machine intelligence, human health, and long-term futures. Here we explore how living systems are engineered, how intelligence emerges from cells to societies, and how we stay grounded while working at the frontiers of science and technology. I'm your host, Corey Smith, and today's conversation sits right at the heart of that intersection. Today, I'm joined by Frank Lee, founder and CEO of Stately Bio, a company tackling one of the most fundamental bottlenecks in cell therapy: how to measure and understand living cells without destroying them. Frank's path to biotech is anything but conventional. He's a Stanford-trained computer scientist who helped build large-scale machine learning systems at Palantir, Google, and Google X, including foundational work on sparse tensors at TensorFlow. He later led machine learning at Calico, Alphabet's aging research company, where he pioneered the use of computer vision and high-content imaging to extract biological state from microscopy data, work that placed his team among the top performers globally at NeurIPS. At Stately Bio, Frank has brought these computational instincts into biology, developing a patented machine learning platform that enables label-free, single-cell characterization while keeping the cells alive, a capability with profound implications for stem cells, cell therapy, and regenerative medicine. In this conversation, we'll explore how machine learning becomes a scientific instrument, what cell state really means, how software thinking translates to living systems, and what it takes to build a company at the edge of biology and computation. Frank, welcome to Galaxy Balance.
Thanks for having me, Corey.
Absolutely. So I want to start with a bit about your origins and your career arc to where you're at now. Your career spans many great companies: Palantir, Google, Calico, and now your own startup, Stately Bio. What thread connects all those chapters?
Yeah, it's really interesting. So I'd long been interested in computers from a very young age. Was programming very early on. And I think the thing that really clicked for me was in college when I was at Stanford. I stumbled into a class from Drew Endy, actually, one of the pioneers in synthetic biology. Very inspiring, very motivating for me to see the threads around taking engineering-oriented approaches and being able to apply that to science was really just a novel idea to me at the time, and something that I thought was incredibly exciting. And so I wanted to fuse my passions in computer science and programming in general with applications in the scientific fields. My history actually includes a little bit, prior to my time at Palantir, I'd co-founded a mobile payments company back in the day. So, there was always this entrepreneurial spirit that was part of my upbringing. And really connecting all of those dots is how I ended up here, founding a company at the intersection of machine learning and biology and, being a startup founder.
Yeah, it's great to be inspired by those early teachers and influencers. They can really send us down a long path. And is that the moment that biology clicked for you? That was actually my next question was there a moment either then in that classroom or later as you went through some of these positions that you really wanted to shift into more biological programs?
Yeah, I mean, the only reason I took the class was because I was already interested in biology, right? I don't think it's fair to say that that's where it all began. But I do think that there was a moment, somewhat of an aha moment, if you will, where I saw the same kinds of software engineering principles, abstraction, modularity, designing interfaces, and the idea of being able to take those same kinds of approaches and apply that, maybe somewhat fancifully at that point in time. Remember, this is the pre-CRISPR days, to synthetic biology and being able to control or program cells. It was just a really fascinating idea to me, and one that I felt was deeply powerful. Right. With regards to my interest in biology, I think there's always been this fascination on my end with machine learning, artificial intelligence. Like, how does life and intelligence really come into being, and getting to the root of what it is that makes us human and what it is that allows us to do things that rocks can't do, and other life forms seem to be less able to do. I think it's really somewhat of a deep philosophical question almost for me. What is it about that nature of intelligence that we could actually start to understand and then emulate, and then maybe one day build super intelligent beings?
I found that question and interest among a lot of the people I've interviewed, and that understanding of intelligence and what lets us ask that question. It's a deep philosophical question that drives lots of us. So what are your thoughts after studying it for years? Do you think it's something entirely intrinsic that comes about from the brain manifesting, or where does that deeper consciousness, not only of rocks, which may have no consciousness, but animals or even primates that have consciousness but not this deep ability to reflect and create new technology?
Yeah, I can certainly share with you my personal thoughts. I don't profess to be a philosopher in this regard, and there are a lot of different kind of perspectives on this. The way that I see it is life is really this state of motion, if you will, and what we as living beings are able to do is fight against entropy. And so intelligence is really more, in my perspective, something that emerges as a result of this desire to make sense of this very entropic world that we live in, this broader universe that we live in. And so from my perspective, being able to get to intelligence is really just one small stepping stone on the road to arguably many layers of super intelligence beyond whatever we could fathom today, that we just happen to be at that critical point in time where we happen to observe our current capabilities. But I have full faith that as technology develops, as perhaps super intelligence and then super intelligence and so forth gets developed, we'll be pushing the frontiers of what we as a society consider to be intelligence. And I think that that's a really exciting thing to be on the cusp of.
Yeah, you mentioned fighting entropy. It's an interesting concept that cells do this, but obviously not the whole system. They push the entropy out to the environment. Have you read Isaac Asimov's The Last Question?
I have not. I think it's going to need to be on my reading list here.
I would add it. It's exactly that. It's written probably close to 80, 70 years ago now, but it's essentially that is the last unsolved question. It gets into artificial intelligence. This is well before anything advanced. Yes. And it tracks mankind from Earth to colonizing the solar system and the galaxy, and this artificial intelligence grows with man, and it eventually answers all of men's questions except this one. Can we reverse entropy? Can we relight the stars? And it is a great journey, a great story. It's a short story. It's a very quick read, maybe 20, 30 minutes. I'd highly recommend it, and it has a surprise ending for you.
All right, on my list.
Yeah. So just a little bit more about your story. Some of these companies, we've discussed it a little bit, but what inspires you to be where you're at now? Could you walk us through some of the key decisions along your way and how you made those decisions and how they got you to where you are?
Yeah, so maybe just walking a little bit chronologically. I started out at Palantir really, I think, motivated by the mission of the company, and I think that is what compels me also today. I've always been a very somewhat idealistic, I would say, and a mission-oriented person. And to me, the idea of being able to build software that could help understand very complex data sets, intelligence packets, whatever. That was what motivated me to join Palantir. And at that point in time was actually when I first saw the ability to couple the more traditional forms of software engineering with what was happening in the broader world around the deep learning revolution, and being able to use machine learning and artificial intelligence to go and understand those kinds of intricacies in data sets much more profoundly and much more automatically. That's when I transitioned actually to Google X, working on a machine learning moonshot. This was the moonshots arm of Google at the time. This is pre-Alphabet. And that was really where I first saw the ability to take these kinds of machine learning technologies and actually build them into tangible products that people would be using. Now this particular moonshot remained within Google and later got folded into through a series of reorganizations and became the precursor to the modern day Google Assistant, which obviously has since been leapfrogged by several folds with Gemini and so forth. But that was very early days machine learning, right? And I was actually very lucky to be part of a team there where through some of these reorgs, I ended up as part of the Google Brain team. This was the place where all of the top machine learning researchers had been assembled, in the 2014, 2015 era. I was an early developer on TensorFlow, as you had previously mentioned, which is a big machine learning framework that came out of Google. And I was personally part of a team that was dropped in to help other product areas within Google figure out how to deploy machine learning in production. So not only was I building infrastructure on one hand, but also figuring out from the users of that infrastructure what were the features that were necessary. And I had a blast. I had personally got to work on Gmail and ads and Play and YouTube. It was a very expansive experience. And for me, the big opportunity came when I saw how impactful these kinds of technologies could be at scale in the consumer side. And that kind of itch around, how can we take these very powerful technologies and bring them to bear in the life sciences field, which I thought would have profound implications for humanity. We're all biological beings, after all. And so that's when I made a very conscientious focus and switched to Calico to really focus on taking these kinds of technologies and applying them in this domain. So a bit of a circuitous path, but for me, I think it all sums up into hopefully a coherent story arc around why these kinds of technologies really motivated me ever since the beginning and have continued to motivate me since.
Interesting. And a little bit more into Calico. What do you think, how did this shape your thinking about aging, disease, and biological complexity? Our last guest was Chris Bradley, aging company, and I have some other aging experts coming up. So I want to get your perspective more from the computer programming, but working at this big company, Calico.
Yeah, I mean, Calico was a fabulous place. It was a really interesting question, which is what attracted me, I think, to joining there in the first place. Aging is a very obviously important concept that affects us all. I don't think anyone has ever been able to fully define what aging means. And there are different computing frameworks for how to actually go about defining that in a way that could be objectively measured, right? For instance, chronological aging obviously is fairly objective, but I don't think that anyone purports to be able to reverse time. And so I think people mean something different when they talk about trying to slow aging or maybe even reverse aging. So how do we think about aging, scientifically in a way that could be experimented on, tested, validated? Those are some of the key questions that I think were really challenging. A little bit of a prelude to what we're doing at Stately Bio. In my time at Calico, one of the technologies that we were working on was applications of computer vision to the analysis of microscopy images of cells. And in a somewhat unbiased way, we were trying to get at this notion of how could you phenotypically characterize biological systems, in this case cells, to be able to give you the kinds of features that perhaps you could use to characterize some notion of age-related state about that cell. And I think that that's a really foundational question at the end of the day, is given that we have no clear objective definition of aging, how do we go about maybe letting these machine learning models just look at data and infer from the data the relevant features for defining age in various contexts. And so that's really how all of this interest in computer vision applications to cells really got started. With regards to aging, I think it's a really interesting question. Obviously, there are a lot of moneyed interests who are interested in tackling this in various ways. For me, I think we are still fairly far from understanding what it means to be aging, to then be able to course correct for it. And I think there are very practical challenges with regards to how one would run trials, on the order of the lifespan of a human to actually get at any potential, aging-related drug. But I think that those are all challenges that will be overcome in due time. I do have continued to have a lot of interest and support for the aging field.
And there's a lot to discuss: longevity, escape velocity, or essentially, if you live long enough, you could live forever. What are your thoughts on that?
I don't know that if you live long enough, you would live forever. Forever is a very long time, right? So I think the statistic, don't quote me on this one, but there is a background rate of accidents or other incidents that would occur. So even if some notion of natural aging, whatever you want to interpret that as, were to be able to be completely eliminated, there would still be an expected life expectancy that is not infinite. And far below that. The estimate that I had heard was somewhere on the order of less than 1,000 years, given the current background rate of incidents. You probably know the statistic better than me.
Yeah, not immortal as in impervious to knife attack or bullets, but stopping natural disease-related biological causes of aging.
But those are I think that's the interesting question, is like, well, how do we define what we consider to be natural and unnatural, right? And at some point, it will become, I think, very hard to tell the difference. So to give you an example, right now we've already figured out how to, as a society, eradicate so many of the virulent pests that previously have affected and severely limited life expectancy, right? That, you could argue, is a unnatural way of meeting one's end. But at the same time, it is part of this broader ecosystem of the biosphere that we live in. So how do we actually think about what it means to be naturally aging? Does some notion of say mental health factor in, and how does that factor in to different definitions of death, even brain death versus heart arrest? Like, these are all, I think, real questions that will become more and more prevalent the farther and farther we are able to go and push the longevity up.
Yeah, you bring up a good point of early childhood disease, go back 500, 1,000 years. There's such a big mortality curve right around there. We've essentially pushed almost all of that down to the right, and how can we extend otherwise healthy conditions beyond that? I think it's a really interesting question. I think there are lots of advancing frontiers. It's a very fascinating field to watch. And do you follow anything personally to extend your lifespan, from a mental practice or physical practice or any therapeutics or supplementation?
Yeah, not in terms of therapeutic supplementation, but in terms of dietary schedule. I do practice kind of caloric restriction, and I do think that is something that probably is one of the more scientifically validated approaches to potentially having a positive effect. We see that in various animal models, and in general, I think there has been, at least for me personally, no obvious downsides to doing so.
And do you follow intermittent fasting or just general reduction throughout the day?
Yeah, I have, I typically don't eat, for instance, breakfast, lunch, and dinner, and then that's it.
Yeah, just lunch and dinner?
Yeah.
And when did you start this? How sustainable do you find it, and what have you noticed as you've done it?
Oh, I've been doing it for many years now, maybe even inadvertently back in my college days because I would not get up in time for breakfast. Since then, I think I've generally kept this up for, yeah, over a decade at this point. I
Yeah.
don't think it's been, maybe I'm just so used to it now that I don't see any alternative. But
Yeah.
yeah, I don't have any problems.
Yeah, I've been practicing this as well. My first blog post was on this topic. I think the science is very sound. And if you look back to how humans used to live, we did not get 3 square meals a day. I think there are
Sure.
certain energetic metabolic cycles, and having constant food throughout essentially almost 24 hours for some people who have a late night snack, it's very unnatural, and we don't have the systems to fully combat and address that.
Yeah, absolutely. Yeah,
Okay.
and I think that there are these kinds of maybe interventions, like you're saying, whether chemical, therapeutic, or just in terms of dietary or other kinds of practices that we could find to improve our overall health and kind of health span. I don't believe that we are there in terms of the peak, right? I think that there is still a
Yeah.
long road to go. And I fully believe that we are just at the cusp of understanding how to even define the problem so that then we can study it further.
Yeah, I agree. All right, great introduction. I now want to segue into Stately Bio. So if you could
Sure.
just, what is the premise? What's the challenge that inspired this, that you're overcoming? And talk a little bit about the creation of the company and the original idea.
Yeah, so Stately Bio, we develop machine learning technologies for understanding and characterizing cells in a way that is non-perturbative, right? And what I mean by that is not having to kill cells, not having to fix cells, just taking, excuse me, white light microscopy images of cells coupled with machine learning analysis to be able to understand what a cell is doing and how to eventually control those cells. The way that this got started was actually, as I alluded to earlier, in my Calico days, my team participated in a contest held by Recursion at the world's largest machine learning conference in Europe, where they had taken images of cells treated with 1,000 different siRNA perturbations. But they didn't tell you which ones they were, just siRNA number 1, 2, 3, all the way to 1,000, and asked you to take a new image that was of cells treated with one of those 1,000, but they didn't tell you which, and they asked contestants to build a machine learning model that could take such an image and identify which one of 1,000 different perturbations it was. It was set up deliberately like the ImageNet task in machine learning, if you're familiar with that, 1,000-way classification. And when we entered into it, we thought, yeah, this is like maybe a 50% accuracy would be an amazing outcome, right? To the eye, there was really very little signal that you could see about these cells. By the way, they were using their kind of cell painting approach, so multi-channel images of dyes that would stain for different parts of the cell membrane, nucleus, nucleoli, so forth. And what ended up happening in the course of that contest really surprised us. We ended up placing second and got over 99% accuracy at the very end. And it was one of those kind of aha moments for us that we realized, there's a tremendous amount of signal in these images. And moreover, our priors as to how much signal there was completely off. And so that was something interesting to us. Anytime you have those kinds of big discrepancies between what you expect and what you're able to accomplish. And so that led us to double click a bit more. And so that kicked off a very interesting line of research at Calico. Now, getting back to Stately Bio, the reason why I founded this company was, despite all of the very exciting work that was being done there, one of the key limitations was still that you had to fix cells in order to capture these kinds of images. And as we all know, biology, life is living, and it's a little bit ironic that we're studying dead matter at the end of the day in these kinds of images. And so the question there was this kind of thought in my head around, could we actually take similar approaches, but do away with the staining, right? Do away with the fixation. Keep cells in their natural state and with minimal perturbations be able to read out those same kinds of very high signal present in those images. And that's really, what's led to the birth of this company is the realization that, in fact, you can do that, and quite effectively so, and with profound applications, in our case, towards stem cell differentiation and applications on the therapeutic side, because now you can actually start to monitor how cells change over time, right? How and experiment on those cells to develop better protocols, for instance, to improve differentiation of stem cells into desired high-value therapeutic cell types. That's something that would be incredibly hard to study if every single time you wanted to take a measurement on a cell, you had to kill it. And so being able to do so in a non-destructive fashion, I think, has really opened up new doors for us on the therapeutic development front, which we're pursuing now, but then also with regards to just a host of applications more generally. At the end of the day, this is not specific to one cell type or one biology area of interest. And so we do view this as a platform technology of sorts that we think can be applicable for therapeutic applications and non-therapeutic applications as well.
Excellent. And in your early intro, you mentioned to Google learning what it takes to make a software accessible to all, being part of Gmail, these other products that really billions of users ultimately end up using. What part of that do you incorporate into Stately Bio? Is there I don't think there's an open version, but how many users do you have, and how are you making this software accessible or empowering to others?
Yeah, we're thinking about this deeply by integrating the technology development internally with the kind of power users, if you will, of this technology ourselves, right? And so by being able to integrate that with wet lab experiments that we're running ourselves, we can actually innovate quite rapidly on both how do we actually develop the technologies and also how do we actually use this to do science in a different way. I'll give you a very concrete example. When we think about stem cell differentiation, there's a tremendously large number of knobs that you have to turn. Different reagents being introduced at different times, different concentrations, different combinations, different durations of exposure, and all of that combinatorial mess is just too large to exhaustively search. Now, at the same time, being able to run screens to find better protocols is something that is going to become enabled when you can have real-time readouts of how well your various conditions are performing. And so we've actually run a couple of screens where before the first experiment even finished, we already knew which ones were likely to be the conditions that we wanted to follow up on. And so we were already able to start the second experiment to validate those prior to the official conclusion of the first one, right? And so this is just a new paradigm for how you can think about compressing timelines when you do discovery and really rethinking how we can do science, right? Another example that I'll share is we've had some work with collaborators on being able to forecast into the future how differentiation outcomes are going to be. So because we can keep the cells alive, we can actually take a look at how cells are behaving early on in the differentiation and correlate that with outcomes far into the future. And when we can build these kinds of what we call forecasting models that can predict based on early time points what's going to happen in the future, you can start to think about designing your experiments with a much tighter iteration cycle. You start to have different trade-offs around how willing are you to tolerate false positives versus false negatives if you have such a proactive ability to read out cell states directly in your experiment. And so we're very bullish on this idea that these are foundational technologies. Right now, we're developing them in-house because I think that the iteration cycle is going to be much faster for us to be able to develop these technologies. But then, we do view this as a platform technology that will, in the fullness of time, be something that we think is going to redefine how we think about monitoring cells in a non-destructive fashion.
Excellent. And there's been so much promise since Shinya Yamanaka reprogrammed iPSCs, and I think there's been some disappointment in how long it's taken and certain stumbles that have come across the way in terms of cost or not clear regulatory path. How do you think your technology could address those two main ones of cost and regulatory path?
Yeah, well, cost I think is twofold. One is the length of time, and then the nature of success rate, if you will, and all of that is kind of quality. The length of time is one that I think many protocols today can be substantially shortened. And we've already seen that in our own hands. But then with regards to a mix of yield, purity, quality, and so forth, that's the other component to cost, is how many cells do you have to make in order to get the cells you actually want. I think that that's a really big area for improvement in these protocols. And that's exactly well suited for these kinds of screening approaches that we're taking, where it's essentially an objective function that we're trying to maximize. And we have a real-time measure of how well those cells are hitting those goals, right? So once you can actually start to maybe adopt that kind of machine learning-ish mindset to thinking about biological systems, I think a whole world of possibilities opens up. With regards to regulatory, I think that there is obviously a lot more work that needs to be done to have the first iPS-derived product be approved by the FDA. I think would be, everyone is kind of trying to make that happen. I do think that there are certain related regulatory implications, though, beyond just the iPSC use. One is this idea, especially in manufacturing, that right now we have, essentially we follow a SOP, right? There's a set of constraints around what the product needs to be. There's a set of instructions around what we do and how long we do them. It's all fixed in stone. And I think there's emerging, a realization that in fact having more adaptive, more reactive, and more proactive technologies to monitor manufacturing processes as they're occurring, and then be able to change those protocols in ways that will allow you to have a better outcome. Those kinds of process analytical technologies, I think the FDA has expressed a keen interest in expediting. And I think that that's one area where we're obviously very interested in expanding into. These kinds of technologies are perfect for the manufacturing suite, where you don't want to touch the cells. You don't want to perturb them. You want to keep the cells alive. You want to be able to reuse them. And being able to get real-time readouts about your biological substance of interest is going to be incredibly powerful. Now, there's a regulatory challenge there, because now you're talking about introducing a statistical model into the characterization of cells, and maybe even the QC and release criteria for cells. Like, how does the FDA and other regulatory bodies think about that? I think that's a really interesting open question. But with the rise of other applications of machine learning in diagnostics, for instance, and other approaches, I do believe that the regulatory frameworks will be there for being able to apply that to this domain. And I do think that that will address the cost issue as well, because now you're able to have much fewer unsuccessful batches, much less waste.
Yeah, I think it's good that the FDA is open to this, but as you mentioned, under current SOP guidelines, anything of analyzing or changing the SOP midstream based on the data, that framework is not yet there, but hopefully you and others can help provide the benefit to that. And one big advantage of the non-destructive testing is everyone else takes that subset of dead cells, as you say, and that's really what they do the QC on. None of those actually go into the product. And with your live imaging system, how much of the total cell population will you analyze?
Yeah, it really depends on the assay that you want to run, essentially, right? There could be, if you really care to analyze all of the cells, you could, right? Oftentimes, I think a more kind of statistical sampling approach might be sufficient for your needs. Again, it depends on kind of your limit of detection requirements and so forth for being able to identify, the positives of interest here. We can go, because imaging is inherently single cell scale, right? We can go to as few as 1,000 cells or so that you can fit into a tiny microliter drop, to as many cells as you need, just spread out over a longer scan time. And so being able to dial that in and out is really one of the, I think, advantages of the approaches that we're taking. Similarly, on the screening side, with regards to temporal resolution, we've developed automation solutions where we can dial in and out how much temporal resolution we need to be able to actually characterize the trajectories of these cells. So we can trade off throughput for temporal resolution. We can't create time at the end of the day, but we can divide that time in different ways across either imaging, one set of biological conditions, very, very high resolution in the time domain, or we can spread that out over a more sparse interval, but spread out over a larger set of conditions.
That's what I was interested in. So essentially imaging every day over the 10-day differentiation period, for example, or perhaps culturing it for a number of hours or minutes to get longer term in a short time. I think stem cell differentiation is beautiful when viewed at that video. It makes a very powerful statement in presentations when I see it. Do you use both of those?
Yeah, absolutely. And so there are, ranges of timescales from the order of, every few minutes, consistently throughout the entire course of a stem cell differentiation run that we're doing, to daily or even less frequently than daily kinds of images. Again, depending on the needs of the scientific question that you're pursuing, right? And that's really, I think, the beauty of this approach, is that it can be easily adapted to figuring out, based on the needs of the scientific questions that you're trying to answer, what is the right trade-off between throughput and temporal resolution.
And speaking of making therapeutic cells, on your website, I saw you have featured albumin and CYP and urea production. These are clearly features of liver cells, of hepatocytes. Was this just used as an example, or is there a particular interest in that as one of the therapeutic options? And you showed some pretty compelling data that over the standard protocol you can improve it. What improvements are made? Is it just through imaging and selecting, or what gives it that difference when run through your differentiation or analytics system?
Yeah, so the way that we deployed our technology in the context of the hepatocyte example that you're referring to, and we've shared this at public presentations as well, is through exactly that kind of screening that I mentioned earlier, right? We were able to run through a, I would say, medium-sized library of different perturbations in combinations in different timings, and search through that space much more efficiently using our technologies, to then find protocols that our machine learning models had predicted would improve hepatocyte maturation in this case, as quantified by albumin and CYP3A4 and urea, right? And so being able to have a machine learning model make those kinds of quantitatively accurate predictions directly from imaging allowed us to then rank order what were the most promising candidates. And then, through several iterative rounds, we were able to arrive at progressively improved protocols that allowed us to kind of share the results that have been posted publicly. So at the end of the day, the way that we're thinking about using technology in this case was as an R&D screening tool, really, as a way to get real-time insights into this very dynamic, very hard to understand differentiation process, and to make sense of that, to be able to then suggest tangible improvements to those protocols that can then be validated on ground truth assays like these functional assays that you were referring to.
Mm-hmm. Excellent. And I think hepatocytes are a great example, because some cell types you might need just partial function to be therapeutically useful. Hepatocytes have a very wide range of functions, from drug metabolism, protein biochemistry metabolism, whole host lipid uptake and release, and for certain diseases you might only need a subset of those. For iPSC-derived therapies, how do you think of making an exact cell type with all functions versus limited functions? Do you think there's room for both, or how are you thinking about it at Stately Bio?
Yeah, I mean, I do think there's room for both, certainly. You have to zoom out, of course, and think about this also in the context of alternative non-cell therapy approaches to tackling those same diseases, and whether cell therapies are ultimately the right solution for some of those, say, metabolic disorders and other diseases of interest. I do think that what we've developed here is really a flexible kind of capability to model hepatocyte maturation as a whole. Mm-hmm. And in our hands, we've seen that when we make improvements to maturation, that is a holistic improvement across the board in all of these different functions. Mm. That gets us closer to, I guess maybe backing up here, the pretext here is that the existing differentiation protocols are produce incredibly immature cells, on the order of 2 to 3% functionality in many cases compared to primary human hepatocytes. And so being able to quantify and predict that degree of improvement on this hepatocyte maturation axis holistically. That's something that is, I think, very attractive about being able to use imaging. Now, there are certainly applications like you were alluding to where maybe just one of those parameters matters more in a clinical context, right, for a particular disease. And that certainly is going to be the case, I think, more generally. And so that's fine. That's just a different objective function, if you will, to then try to maximize in the course of screening. And so I think that this is a very powerful paradigm. With regards to your question around how do we think about B cells in particular, I do think that having, like you said, holistically more functional, mature hepatocytes is going to be a big win for the field. One of the main challenges for kind of hepatocytes as therapeutics is that it's incredibly difficult to source them from typically discarded livers that were not suitable for transplantation in their own right. But then also, you need those cells to perform so many functions, right? And so that is, I think that is where having improved maturation across the board is really going to be the key needle mover here.
And there are many functions beyond therapeutics for hepatocytes. I think for drug screening, modeling, the liver is so active for small molecules and other compounds that have a lot of uses beyond therapeutics. And like you mentioned, one challenge is not enough primary cells. But even with good primary cells, they don't seem to have much therapeutic effect from the research I've seen. I think for targeting a cell type, you really want something that has a strong effect with the primary cells. If the primary cells don't already do the job, I think you'll have a hard time making an iPSC-derived therapy actually work.
Mm-hmm. Yeah, I think that one of the key challenges working with primary hepatocytes is that they start to de-differentiate very quickly in culture. They lose their function on the order of, I've heard, hours. Sometimes, people have been able to push it out to days, but not very long, right? Yeah. And essentially they're use and discard, because after that point, you can't go back to the primary state that they were in. And so that makes it very challenging, especially if you have to source them from different donors, which have obviously a lot of donor-to-donor variability. That makes the use of those kinds of primary cells for toxicity testing, disease modeling, much more challenging. And that's where we think actually having a reproducible manufacturing approach using IPS could actually lead to, even if it just simply matches those primary human hepatocytes, that would already be a huge improvement over the status quo.
Excellent. And one thing that's heavily mentioned on the website and your technology is using those ML image approaches to classify cell state. Just for the audience, clarify what you mean by cell state and why you think that's important to get to with your analysis.
Yeah, it's a great question, and actually, that's why the company is called Stately Bio. Also, is a nod to this realization that cell state is what I think of. And different people will use the term loosely to mean different things, but the way that I think about it is, imagine if you had some kind of approach to simulate a cell altogether, right? The totality of the parameters that would be used to describe the cell, that would constitute its state. Now, that is an abstract notion. We cannot ever fully describe all of the kind of parameters that would be needed to fully simulate a cell. But what we can do is capture projections of that very high dimensional kind of state space in lower dimensions. One projection could be using transcriptomic approaches, for instance, trying to understand what are the set of transcripts that are actively being produced by the cell. Another set of approaches could be using protein expression and like marker-based analysis. I think another approach is using imaging to get at a notion of how does the form of what we see under a microscope, how does that form relate to the function of that cell? Right? And all of those are different ways of interrogating this very abstract notion of cell state that I think are imperfect in their own right, but collectively, each has their own advantage and disadvantage, and collectively, they give us different views of what it would mean to be able to describe a cell in its totality. And so the way that we think about it is, being able to model cells at the end of the day and be able to predict what they're doing at the level of granularity that we care about, which is oftentimes functional assays or other kinds of clinically relevant endpoints, like that's ultimately what we want to build models to be able to do. And being able to achieve that end using readouts that are highly scalable and kind of effective for machine learning is really what this company is focused on.
Wow. And one focus has been on pushing from that pluripotent state into the desired target cell type. Do you also look at the importance of, especially for manufacturing, maintaining the IPS state for the cell bank and those original precursor steps to get enough cells, pluripotent cells?
Yeah, absolutely. I mean, that was one of the very first applications of the technology, was to be able to identify, are these cells still in a good pluripotent state versus not? I think that's just one specialization of this broader general framework that I was describing, right? Where you can identify, are these cells still pluripotent cells or aberrantly differentiated cells? And then being able to go the next step, which is now there is a direction of travel around, getting from state A to state B. How do we actually map out and quantify the degree of progression from one cell state to another? Kind of the mental analog here that I would like to share is, imagine you have a map that you're trying to locate where cell states are on this map, right? Right now, we don't even have the map, so we're trying to build out those kinds of maps. But then, moreover, we're trying to build the understanding of the road networks that will connect them, so that then we can provide, in a more engineering-oriented kind of design-based approach, a set of directions for how to get from state A to state B, right? And we think that those kinds of maps and this kind of understanding of biology is going to be enabled through these data-driven approaches coupled with computational machine learning.
Exciting. And you mentioned, I think, the most abundant data, at least in terms of sheer amount and publication, is transcriptomics, from single-cell transcriptomics and just general bulk RNA-seq. Do you ever, using your map analogy, map them, like an electrical route and a water route, and then the surface image? Do you look at these underlying molecular data and use that to help train it, or at least check it afterwards? Or how do you see integrating multi-omics with imaging?
Yeah, I mean, I think that's the big question that everyone is trying to figure out how to actually integrate. I think one challenge with the transcriptomic data is that even if you do, for instance, single-cell RNA-seq, the single cells that are being read out there are not the same single cells that you could collect via imaging, right? So that kind of one-to-one correspondence is lost. And so oftentimes what we end up preferring are approaches where we can interrogate cell states while preserving kind of the imaging based approach to allow us to have one-to-one correspondences be maintained. Obviously, immunofluorescence staining and other kind of marker-based approaches lend themselves nicely to that. Also, FISH and other, more recently, spatial transcriptomics, I think, is a very exciting direction to be pushing in, where I think being able to draw, again, that correlation between form and function is going to be incredibly important. And being able to use those kinds of technologies to develop models that can, again, analyze non-destructive phenotypes and be able to predict what those cells are actually doing and model those cells. I think that that's really the next frontier.
Yeah, I was going to bring up spatial transcriptomics as well, like FISH-seq, fluorescence in situ sequencing, clearly still destructive, but at least you could get image data and the molecular data of what transcripts might be there.
Yeah. The ground truth data oftentimes is going to be these kinds of destructive approaches because that's the bulk of what exists today, right? But I think being able to draw the connections between the non-destructive imaging that can happen in parallel with those destructive approaches, that's a stepping stone toward connecting the bridge between the world of imaging and the world of molecular data that we would have a ton of data around.
Excellent. Great description of the company. And so if everything goes perfectly, what's your vision for the company? Where can you be in a year or 5 years, or where's your next goal? And if really everything works, where is Stately Bio going to be?
Yeah, I think the grand vision here is hearkening back to my earlier comment around synthetic biology and being able to do more engineering-oriented kind of design of cells, right? Being able to understand how to measure cells, like that's really the technology that we've developed right now. I think being able to harness that understanding to then control cells is what we're actively developing. And I think in kind of the glorious future, as we actually apply those kinds of understanding of control to therapeutic as well as non-therapeutic applications, I think there will be a host of gainful applications for society. On the therapeutic front, it could be cell therapies, regenerative medicine. On the non-therapeutic front, it could be more engineered microbes or other kinds of non-human cells that are producing cells. It's kind of like a biomanufacturing application. I think that there are applications well beyond even just therapeutics and biomanufacturing in the realm of fertility, cell cultivated meats. There's a whole broad spectrum of applications here. We're right now very focused on therapeutics using human cells and iPS, primarily because we think that that is the biggest opportunity near term, where our cells, our technologies and our cells will allow us to make a significant impact and contribution to the world. But in the fullness of time, I think that there's a very expansive view. And that's why I think about this company as truly a platform technology company at the end of the day. We're not just trying to build one therapeutic using this technology. We really think about this technology as enabling, hopefully, a whole field of regenerative medicine and things beyond that as well.
And what are some of the greatest challenges to get there that you're dealing with today and that you see in the future?
Yeah, I mean, obviously there's this tension, I would say, between building platform and building a product. I think that this is something that we are not the first ones to come to this. I actually find it incredibly empowering to be able to use the fact that we are actually developing our own therapeutics to motivate the design decisions around what are the aspects of technology development that we actually need to make in the platform. Right? I think that that's actually paid dividends in spades. But there is that tension around, well, how do you balance resource allocation, especially since developing therapeutics is very expensive and oftentimes mismatched in terms of the speed at which that goes versus the speed at which technology development could go. So how do we actually balance those needs in the company? I think that's one challenge. Another challenge, I think, is this broader idea around, I don't think there's been a very, maybe I would say, there's not a clear playbook to follow for how to build a platform technology company of the sort that I was describing in this space. I think people have taken different approaches. I think there certainly are elements to learn from each of those. But how do you actually build a sustainable kind of business around this platform technology while developing therapeutics? I think is something that we're trying to write the playbook for, right? And that's something that I think is something that I think about a lot.
Excellent. And you've talked about a lot your ML approaches to the actual vision and the images. Are you incorporating any LLM or other AI approaches in the experimental design or interpretation of results, or how do you see that broader ecosystem in advance? And as you discussed earlier in the conversation, scientific superintelligence, or what comes next, how do you see this broader ecosystem merging with your specific company and technology?
Yeah, I think I'm very excited about the kind of co-scientist type approaches that people like DeepMind or companies like DeepMind and others are developing, which allow you to reason. They're not just encyclopedias, right? They're not just being able to look up past references and do all of that very kind of tedious work for you very quickly, but they're actually able to reason about why did something work or why did something not work. And be able to provide, hopefully, one day, more calibrated probabilities around what are the likelihoods of different alternative explanations as part of that experimental design loop. I think that that's incredibly exciting. We're using them peripherally right now in that fashion, not so much to directly look at images and tell us what is going on there, because I think that that's not their strong suit at this point. Mm-hmm. But I do think that being able to integrate the holistic body of scientific knowledge and then be able to reason about, especially in the context of very specific questions that a scientist at our company might have, what if we did this, what if we did that, how is that likely to impact the say, the forecasting predictions. Those are kind of the next level of machine learning applications that I think will be incredibly exciting to build.
And where do you see the role? A lot of people hear this, even other scientists. I talk to some of my mentors who are since retired and describe some of these things on the front for scientific superintelligence. They're like, are there going to be any bench scientists? What's the role? So where do you see jobs and employment and future scientist roles and development with these advancing technologies?
I'm not very concerned about job security for the scientists there. I think that there's a lot of work still to be done. There's obviously the very physical stuff that these digital tools cannot, at least in the current day, manifest, right? Physically handling liquids with solids and being able to move reagents around, there are some automation for that, and I think that I do expect that to get better. But that's still, I think, a main bottleneck with regards to hands-on time, right? With regards to the design of experiments, I do think that that's one area where current scientists will become incredibly empowered to be the best versions of themselves by being able to tap into this whole body of knowledge that is really exposed through these kinds of LLMs or other kinds of co-scientist approaches. I think the most important thing for me is the domains in which these kinds of models have really seen big gains. You take a look at mathematical reasoning, you take a look at programming or writing code. These are loops which can be self-verified in the course of training and validating these machine learning models. And I think that biology is not yet at the point where there is that kind of self-validating loop. I think automation, I think being able to standardize on data collection using these kinds of high throughput means, I think will take us a step closer to that. But I think that it will still be some years away before we get to that loop where scientific reasoning itself can start to become automated.
And what excites you most about the future? It could be something very in your field or with your company, but all the advances and everything that you're seeing, what gives you most hope to look forward for in the future?
Yeah, I mean, I think if you zoom out, right, the way that I think about this is if you take a look at increasing levels of abstraction, starting from physics. We've, as a society, figured out how to do all kinds of crazy physics things. Like we can launch rockets and land them very precisely on distant planetary objects. If you take a look at chemistry, we figured out how to make all kinds of synthetic polymers that are never existing, or to the best of our knowledge, never existed probably previously in the universe. And then you take a look at biology, and we're just barely starting to scratch the surface of being able to design and engineer biology. I think that is the next level. It's a very scary thing to be working on, to be fair, because we are biological beings, and if we do something kind of inappropriate, then that would have ramifications for us all. And we don't have right now a good way to kind of control for that. But I will say that it is a really exciting technological frontier to be working on, is this question of how do we actually understand life in various ways sufficient to be able to then kind of advance and maybe even one day create life. I think that that's something that is incredibly challenging and also exciting for me. And I think that as a society, as technology develops, we are starting to build the arsenal of tools that we need to be able to accomplish those kinds of aims.
Yeah, I heard a great quote that our ancestors shaped stone and we will shape the genome or shape life. And that really stuck with me, of great tools and where we're going, and where we're at is just a rock scratching another rock. But where will that refined machine of genetic engineering look like in the future? It's an exciting thing to imagine.
Absolutely.
And kind of segue towards the end, one thing we talk about a lot in this show is science fiction. So could you share a little bit if and how science fiction may have inspired you, and give a recommendation to the audience?
Yeah, well, as I think we touched on earlier, I'm not a very prolific scientific reader, but one that I really did enjoy was the Three-Body Problem trilogy, especially the second book, The Dark Forest. It's one of my favorites. And then more recently, there was a very short, almost like a blog post of sorts, that I saw, Lena by Quantum, that I would highly recommend. It just really struck me and forced me to think about things in a different way. So highly recommend that for your audience.
Excellent, great. I'm a big fan of the three-body problem, but haven't heard of Lena, so I will definitely check it out. And as you just mentioned, I think one of the great things of science fiction gets you to think of things in a new way, or how many technologies, inventions, new areas of science were first thought about, put into books well before we had the ability to get it, and that inspired scientists to get there. And some final closing questions. What one piece of advice would you give to new engineers or new scientists coming into biology very curious to empower them to go about their career?
Yeah, I think in this day and age, I think there's a lot of consternation that I've heard from new college graduates and others of that age. I think it's a very rapidly evolving time for sure on the technology front. I think those that are able to embrace those tools and learn how to use them most effectively will be in the best position by far to be able to make a dent on society at large. And I think one of those directions right now is we're still at the kind of prompt or context engineering stage of understanding how to use LLMs, right? I think that is being able to learn how to use those tools effectively, whether in the computer science fields, more software engineering more generally, or in biology and being able to intersect those two, being able to learn how to use those tools to answer questions that are highly customized to your own needs is a profound game changer for how effective you can be. And so I would really highly recommend kind of playing with these tools, starting to just be immersing yourself in those. I don't believe that they are right now sufficient to replace the activities of, say, professional software developers or certainly scientists. But I do think that technology develops very, very rapidly in this space. And I think that the more that you can learn how to use these tools, the more better set up you will be for the future.
Well, Frank, great conversation. Thank you for joining us today. Really appreciate it. And please have a great day. Thank you.
All right. Thank you, Corey. Pleasure speaking with you.